That's it.
Okay, thank you very much again, Christine, for the kind words introduction. I'll be quite honest with everyone in the audience. When I first heard that I'd be up here giving the Bitter Lecture in Gothenburg at the 2017 meeting of the SEB, the first words that popped into my head were as follows, and I'll play them for you. Hopefully the audio is on.
Fake news. Fake news. Fake news.
So that's what I was thinking when I first heard the news. Well, it clearly turned out to be quite real. And believe it or not, I'm up here today wearing this Buzz Lightyear outfit and in front of you giving the Bitter Lecture. I've been coming to SEB meetings, I guess, for the past 40 years. Not every year, usually every second year I come to the SEB. So I've had an opportunity to see a number of Bitter Lecturers over the years. And it's always been, at least until today, a highlight of the meeting for me. As I've been watching those Bitter Lectures over the years, I have to say I never imagined that I would be up here actually giving the Bitter Lecture. It still doesn't seem quite real. Obviously, it's a tremendous honor to receive the Bitter Lectureship. It's a great recognition. I'm very appreciative. I'd like to thank the Society for Experimental Biology. I'd like to thank those who nominated me and those who supported the nomination. Once again, I'm deeply honored, and it's a great privilege to be here. You know, it seems to me as I've watched Bitter Lectures over the years that many Bitter Lecturers, if not all of them, tend to seize the moment, seize the opportunity to tell the younger generation in the audience, if there's anybody here, some messages, some words of wisdom regarding life. And this may not have anything to do with science or research or whatever, but some words of wisdom. I remember a few years ago, Chris Wood, when he gave his Bitter Lecture, he told us about his recipes for academic success. And I think it was a 10-point plan that Chris told us about. And I'm going to tell you about the first three bullets on that plan, and since Chris won't be able to disagree with me, he'll have no opportunity to say whether this is right or wrong. But the first piece of advice from Chris was never respond to a memo from your dean. The second point was do as little administration as possible, and the third one is do as little paperwork as possible, especially with respect to animal care protocols and health and safety. Well, this was great advice. This is great advice for the younger generation, especially those looking to move universities. I don't really have anything as profound as that to say to you today. And really what I'd like to do is to get on with it and tell you about some of the data that we've been generating over the last several years. But if it is my role to, as sort of an old fart in the field, to pass along some words of wisdom to the younger generation, might be something as simple as this. That is, there's no single linear path to academic success, just like there's no single linear path to success in life, period. We all take a variety of pathways. We meander along these pathways, and ultimately we get to where we're going to. So the advice I would give, I suppose, is to choose that pathway that brings you to the path or into the path that allows you to do the research that you love, rather than choosing that path that brings you into the research that you think you need to do to become successful. So, you know, in my case, I've always loved fish, and I'm proud to say that I'm a fish physiologist. I've always been a fish physiologist. It may not be the most glamorous field, arguably, in the world, but that's what I've done, and that's the path I took, and I think it's worked out fairly well for me, and it's worked out fairly well for some of my colleagues. So over the past 35 or 40 years now, I've studied a lot of fish, and I'll show you some of the species that I've studied here. In recent years, we've taken to study some non-fish species. I don't know if this makes me a comparative physiologist or not, but we've done some work recently on this land crab with a student from Brazil, Tabata Lima. Actually done some experimental evolution on Drosophila and looking at adaptations. And here I mean adaptations in the truest sense of the word, adaptations to hypoxia and hyperoxia as they evolve through multiple generations. And finally, we're able to do some work on this critter here. This is the bacterium Pseudomonas aeruginosa, and we looked at the evolution of this bacterium in a high salt environment. I guess it's a bit of a cliche, but cliches usually are based on truth. And the truth of the matter is, to be successful, you have to be surrounded by a great team. And I've been very fortunate over the years to be surrounded by great team members. And some of those people are going to flash across the screen. Perhaps if you're quick enough, you'll spot your name. I've had super undergraduate students, graduate students, masters, PhDs, postdoctoral fellows, international collaborators all over the world, colleagues. I'd like to particularly give a shout out to two postdocs who are with us today, two super postdocs, if I can figure this out. Melitza Mandek, who's going to be talking tomorrow about hypoxia-inducible factor and hypoxia tolerance in zebrafish, and another super postdoc, Alex Zimmer, who will be telling us about gene knockout models and how they can be used to look at ammonia excretion and sodium balance. What I'd really like to do is to highlight the importance of three people in my life. Dave Randall, who was my PhD supervisor in the 1970s at UBC, Chris Wood, my postdoctoral supervisor, and Katie Gilmore, my all-time favorite collaborator, both in science and in life. In particular, I'd like to thank Katie immensely for the support that she's given me, in particular over the last 5 years where I've taken on more and more administration. Katie certainly helped to ease the load and keep me sane, although, people might argue that that hasn't actually been the case. Katie and I published our first paper together in 1993, and since that time we've published 65 papers together, and I have to say, Katie is probably the smartest person I know. So we've had a fruitful collaboration, but probably the greatest result of our collaboration has been the production of this data point right here. This is our daughter Audrey Gilmore when she was 3 years old, and already at the time she was showing a love for fish. Here she is playing with hagfish slime at Bamfield Marine Station. She's now 14 and showing no signs of slowing down in her love of fish. So I think the future of fish physiology in Canada is probably quite bright indeed. Dave Randall, who won or gave the Bitter Lecture in the year 2000. At least I think he gave it in the year 2000. I actually asked him this weekend when he gave it. He said, I don't have a clue. So I think I got this from his own CV. Anyways, Dave Randall taught me many things, but perhaps the most important thing that Dave taught me was the art of supervising graduate students using the principle of benign neglect. And I don't really mean that in a bad way at all. I think for those of you who know Dave Randall, and for those students of Dave's in the audience, you know that he's got this unique capacity to know when a student is simply floundering and taking on water versus when they're going down and actually drowning. And it's at this point that he picks you out of the water and saves you. And although we ended up a bit waterlogged because of this, I think many of the students who were with Dave over the years ended up all the better for this technique. He also taught me that the two most important things in science were creativity and the idea. For Dave, it was all about generating the idea. And for Dave, the data and the measurement were not that important. And I think if you've ever done an experiment with Dave, you'd agree with that. For Chris, who was my postdoctoral supervisor, who gave the Bitter Lecture in the year 2012, it was just the opposite. For Chris Wood, ideas are a dime a dozen. And for him, it's all about making the measurement. And not just making a single measurement, but making massive numbers of measurement. So Chris Wood taught me how to make measurements. He also taught me the art of assembly line physiology. And so what Chris said, if you can make a measurement on one fish and collect one data point, you can collect data points from tens of fish as long as you're able to stay up 24 hours in a day, which Chris is able to do unbelievably. So between the two of these people, then, what I learned is if you have a great idea and can make a great measurement, you can do great science. And I think that's a very important message. The other mentor who's not up here, we celebrated this weekend at a satellite symposium, is Pierre Laurent. Pierre Laurent had a great influence on my career, and sadly I don't have a picture of Pierre here, who passed away 2 years ago. This photo is simply to say that mentors and their students don't age. Here we are in the Amazon several years ago celebrating the research career of Dave Randall at a symposium organized by Adalberto Val. And we had a lovely time there. Well, this is the Bitter lecture, so I thought I should say a few words about George Parker Bitter III. I'd like to talk about who he was, how he played a key role in the origins of the society that we're all members of, and then his own particular Guinness World Record, which may never be broken. So George Parker Bitter was born into a very wealthy family. He was a marine biologist. He was an authority on sponges. He worked in his early days on plankton. He was also president of the Marine Biological Association. And he was passionate about all things artistic, so literature, poetry, classical art. But most importantly for this talk, he was an astute businessman. In the late 1800s, George Bitter was studying plankton at the zoological station in Naples, and he happened to be staying at the hotel called the Tramontano Beau Rivage Hotel. And because of the hours he was keeping, he was staying up late into the night, analyzing data, and sleeping most of the day. He did not get on well with the management of the hotel. And in fact, the financial situation at the hotel was very poor. So to make a long story short, George Bitter was about to be evicted. And as the story goes, as he was facing eviction, and the people were ready to evict him were outside his door, he shouted. They woke him up, but he didn't get out of bed. He shouted from his bed through the door, put the bloody hotel on my bill. I'm purchasing it. And he did. He bought the hotel, and he ran it profitably for the next 19 years, basically using it as a place for the guests of the Naples Zoological Station to stay. Amazingly, today the hotel still exists. It's a luxury hotel in Naples. It goes by the name the Grand Hotel Parkers, and it features a bar called Bitter's Bar, and it's quite remarkable. What is really also remarkable is that George Bitter was 26 years old when he did this. Well, let's fast forward now to the year 1923, which saw the creation of a new journal in the United Kingdom, the British Journal of Experimental Biology. Now, a critical component of the success of that journal was the formation of the Society for Experimental Biology. So the society was formed to support, in an academic way, the journal, to give it credibility. And George Bitter was one of the founding members of the Society for Experimental Biology, along with Julian Huxley, the Nobel Prize winner, H.G. Wells, and a number of prominent scientists and artists at the time in the UK. But the society ran into trouble from the very get-go. It was not well liked by the other societies that were present at the time, such as the Linnaean Society, the Genetical Society, and the society run out of Cambridge Zoology, the Cambridge Philosophical Society. And one of the real problems was that the Cambridge zoologists did not want to publish in the British Journal of Experimental Biology. They had their own journal, which was called Biological Proceedings. And so this created real problems, and the journal ran into serious financial difficulties. So So that the cash reserve at the end of the first year was only £3, which is about $6. And that's despite having some pretty prominent subscribers, including H.G. Wells, Julian Huxley, Punnett, who of course is famous for the Punnett Square, and the news editor of the Daily Express. By the year 1925, the journal was in serious crisis, and this is where George Bitter came in and saved the day. And basically, what George Bitter did was to broker a deal between the Cambridge Zoology Group, the Society for Experimental Biology, and the British Journal of Experimental Biology, and he started out by buying the journal. He bought it for £150, and then he sold shares to his colleagues, including people such as Julian Huxley, Lancelot Hogben, Francis Crewe, and James Gray. Now, James Gray is particularly important in this story because he was a Cambridge zoologist, and actually George Bitter appointed him the first editor of the British Journal of Experimental Biology once he purchased the journal. And that was very important because it allowed the Cambridge zoologists now to publish in that new journal. Also very importantly, George Bitter saw to it that the Cambridge Philosophical Society changed the name of their journal from Biological Proceedings to Biological Reviews. So it was no longer a primary publication, it was a review journal, again allowing the Cambridge zoologists to contribute to the British Journal of Experimental Biology, which ultimately was named the Journal of Experimental Biology in 1929. So basically what this saw was the formation of the Company of Biologists, and to this very day, the Company of Biologists and the Society for Experimental Biology are closely linked together, largely through the charitable outreach of the Company of Biologists. So what about this world record? Well, in the years 1904 to 1906, George Bitter was studying ocean currents, and he was basically trying to show that the North Sea currents ran from east to west. I think that's what he was trying to do anyways. And what he did was he had 1,000 messages put in 1,000 bottles, and he floated them out to sea. And inside each one of these bottles was a postcard addressed to George Bitter, care of the Marine Biological Station in Plymouth. It basically asked whoever found the bottle to mark the location, to send the postcard back, and they would receive £1 as a reward. And so most of the bottles were found in the first few months, but remarkably, there was a bottle found off the coast of Germany on an island in the year 2015. And the person who found it sent back that postcard and actually received a £1 reward. At the same time, Guinness certified it as the oldest message in a bottle. And it's hard to believe that they'll ever find an older message, although one never knows. So George Bitter was indeed a remarkable man. Okay, so let's get to some of the science. I want to talk to you about the control of breathing in fish, the why, and the how. And basically, the control of breathing starts with the ability of fish to sense their environment. And sensing the environment is termed chemoreception. And basically, it ensures that fish are able to sense environmental change, and that sensation or perception of environmental change gives rise to neuronal responses and hormonal responses that ultimately give rise to a whole bunch of physiological responses that are typically of benefit and serve to minimize the disruptive effects of the initial environmental change. Now, in our lab, we've been studying the effects of respiratory gases on physiological function. We know that there are receptors associated with the gills when stimulated by respiratory gases give rise to a host of responses. We've been looking at ventilation, cardiovascular function, and the secretion of stress hormones. In today's talk, I want to focus exclusively, I want to focus exclusively on two respiratory gases, ammonia and oxygen, and tell you about their effects on ventilation. But before I do so, I want to tell you a little bit about the benefit of hyperventilation. And to illustrate this, I've copied a diagram out of a textbook. This is a depiction of a portion of a single gill filament. So the fish gill is composed of thousands of these filaments, and erected upon these filaments are hundreds of thousands of lamellae. These lamellae are the functional units of gas transfer. They're lined up in such a way that they form channels, and water flows through these channels in a unidirectional fashion from the leading edge of the filament to the trailing edge of the filament. And at the same time as the water is flowing, the blood is flowing in exactly the opposite direction through these lamellae, coming into the lamellae as deoxygenated blood, picking up oxygen molecules from the ventilatory water and giving up carbon dioxide, so that by the time the blood leaves the gill, it's fully oxygenated and depleted in carbon dioxide. Well, I think it's very clear from this model that if water flow is to increase through the gill, and it's matched by a relatively similar increase in blood flow, there's going to be a greater molecular movement of oxygen and a greater molecular movement of carbon dioxide. So increasing ventilation can be used at times when metabolic rate increases, allowing fish to match metabolism and gas transfer to achieve higher metabolic rates. But perhaps what is less well understood is how hyperventilation can actually also increase gas transfer efficiency. So what do I mean by gas transfer efficiency? Well, gas transfer efficiency really reflects the ability of the blood to come as close to equilibrium with the inspired water as possible. So in a highly efficient system, the blood leaving the gill will have an oxygen level which is similar to the inspired water. And it's been suggested, because of the high flow of water, and I think theoretically this makes perfect sense, that if hyperventilation occurs, the efficiency of gas transfer will increase. But surprisingly, few studies have tested this. Master student Vili Tsanava several years ago generated some data which I think are just lovely and show, I think, perhaps as convincingly as any other data, that hyperventilation does indeed increase the efficiency of gas transfer. So what she did was she looked at goldfish. She used an extracorporeal or out-of-body circulation, so she externalized the blood in a loop. The blood flowed through electrodes, allowing her to measure blood gases thousands of points per second in real time. And then what she did was she injected the respiratory stimulant, sodium cyanide, into the buccal cavity at discrete intervals. And you can see that at each one of these intervals, there's an increase in both breathing frequency and breathing amplitude. And let me just magnify the results for you. And what you can see is that at every time when there's an increase in ventilation, there's an increase in arterial PO2. And this is not insignificant. This is an increase in arterial PO2 of about 60 torr associated with each increasing ventilation event. And at the same time, there's a decrease in PCO2 of somewhere around 0.5 to 0.75 torr. So there's no doubt about it that gas transfer efficiency is increasing when the fish is hyperventilating. So the benefits of hyperventilation then are to increase rates of gas transfer when metabolism increases, for example, during exercise, but also to increase gas transfer efficiency when oxygen levels in the environment become limiting, as they would during hypoxia, or when the fish has to minimize the levels of CO2 in its blood, as it might have to do when exposed to high CO2 or hypercapnia. Okay, what we know is that almost every fish that's ever been examined exhibits hyperventilation when exposed to low oxygen, including this the most ancient of extant vertebrates. This is the hagfish. This shows ventilatory recordings from normoxic hagfish. Here you see hypoxic hagfish. And what we did here was we simply sewed a ultrasonic flow probe into the snout of the hagfish and recorded water flow. I want to show you a video. So what we're doing here is injecting dye into the mouth of the hagfish, and what you can see here is the dye exiting the respiratory spiracles. And what was amazing when we did this is the plumes of water that are exiting these respiratory spiracles, despite the fact that this animal is completely quiescent, and by the way has the lowest metabolic rate of any known vertebrate. But clearly the volumes of water are rather amazing. I'm glad this has stopped because there are people in the audience who may be experiencing flashbacks. Air-breathing fish also do it. Air-breathing fish, when exposed to hypoxia, increase their pulmonary breathing. When exposed to hyperoxia, decrease their pulmonary breathing. This is work that was done in collaboration with Tobias Wang, undergraduate student Rachel Lieberman, and Katie Gilmore. And I have another video here, which shows a lungfish air breathing under hypoxic conditions. There you see it going up into the air chamber, taking a lovely breath, and through the beauty of video manipulation, you'll see it take another breath very quickly, as if it was under hypoxic conditions. In fact, this is a normoxic fish, but we just sped it up to make it look hypoxic. Larval zebrafish also hyperventilate. This is very important for my own research because this is our key experimental animal. What we've done here is to inject food coloring into the mouth area of the larval zebrafish. You can see the breathing movements here of the opercular flaps and the buccal, the mouth. You can see the dye flowing through the transparent buccal cavity. You can see it flowing out of the opercular cavity. And what's very interesting is you can see that the pectoral fins are then playing a role in moving that dye along the body surfaces, because the body surface, of course, is a very important site of gas transfer in larval zebrafish. Okay, I want to switch gears now and start to talk to you about some of the research that we're doing in our lab over the last several years. But I just want to bring you up to speed. So in this slide, what I'm going to do is summarize about 15 or 20 years of research that's been done by a number of colleagues working with me: Steve Reed, Katie Gilmore, Richard Kincaid, John McHenry. Everyone knows Bill Milsom, and Tadeo Ranton from University of São Carlos in Brazil. And basically, traditionally, and this goes back probably 20 years ago, oxygen was thought to be the only respiratory gas detected in fish. Well, we now know clearly that's not the case. We know that fish respond to carbon dioxide. We know that the responses to CO2 and O2 are ancient and highly conserved traits. And we also know that CO2 and O2 act as specific chemosensory stimulants. We know that CO2 act on largely, but perhaps not exclusively, external receptors. And we know that fish exhibit plasticity with respect to responses to CO2 and O2. I want to talk a little bit about the external receptors that we now think are involved in sensing oxygen and carbon dioxide. Pierre Laurent and his group in the 1970s and 1980s played a critical role in determining that the external receptors were cell types which he called neuroepithelial cells. This is a landmark paper published by Dunel, Bailey, and Laurent in 1982. And effectively what they showed in this paper was that under normoxic conditions, these neuroepithelial cells were filled with dense cord vesicles. We now know that these vesicles contain serotonin. I'll just magnify that for you. And under hypoxic conditions, the dense cord vesicles disappeared. So presumably they were fusing with the plasma membrane. The neurotransmitter serotonin was being released into the synaptic cleft and interacting with afferent neurons. In 1992, this was followed by another paper by this time the same team, but in slightly different order, and they showed that these cells were serotonergic. Here's some lovely indolamine histochemistry of these neuroepithelial cells, which you can see lined up along the filament epithelium. Well, it wasn't until many years later, in fact, that Michael Johns and Mark Burleson provided direct evidence that the neuroepithelial cells are sensors of oxygen. And they did this using patch clamping techniques. So Michael Johns worked on zebrafish, Mark Burleson worked on catfish. We extended this study to carbon dioxide several years later, work done by master student Xiaohong Qin. And basically what we showed is under patch clamp conditions in primary culture, the neuroepithelial cells would depolarize in response to hypercapnia. Here are the average data shown here. They would depolarize in response to hypoxia. But more importantly, we found that the same cell would depolarize to hypercapnia, would also depolarize in exactly the same way to hypoxia. So a subset of neuroepithelial cells are bimodal sensors of oxygen and carbon dioxide. Chris Wood and collaborators took this one step further by showing that neuroepithelial cells not only sense oxygen and CO2, but also ammonia. So now we have the neuroepithelial cell as a trimodal sensor of 3 respiratory gases. And what they showed was that when exposed to high external ammonia, and they did this through calcium, intracellular calcium recordings, there were 2 types of responses, one which they called type A, a slow increase in calcium, and one which they called type B, which was a very rapid spike in intracellular calcium. Okay, so what I want to do for the rest of today's talk is tell you 3 very short stories. One about Rhesus glycoproteins and ammonia sensing. Rhesus glycoproteins are simply ammonia channels. I want to talk to you a little bit about hypoxia-inducible factor and oxygen sensing, and then if time allows, the role of serotonin or 5-HT in chemoreception. And so let's begin by talking about these ammonia channels and the role they might play in ammonia sensing. So as I just told you, Chris Wood and collaborators showed that external or internal ammonia loading causes hyperventilation in rainbow trout. Gudrun de Broek also showed that dogfish shark exhibit hyperventilation in response to ammonia loading. The neuroepithelial cells appear to be involved. I won't show you the data for that. And we know that the neuroepithelial cells, at least in rainbow trout, express RNA for the Rh proteins. So with all of this in mind, we came up with the following model, that the neuroepithelial cell shown here is able to sense ammonia because it allows ammonia to enter from the external environment through an ammonia channel. In this case, the RhCG1 protein. It can then exit the neuroepithelial cell through a basolateral channel called RhBG. And if the animal is loaded from the inside with ammonia, as might occur after a meal, the ammonia can enter the cell through the RhBG. So that's basically what we were testing. Our hypothesis is that Rh proteins play an important role in allowing neuroepithelial cells to detect ammonia by allowing ammonia to gain access to the neuroepithelial cell. And if that's true, we would predict that translational gene knockdown of Rh proteins will diminish the hyperventilatory response associated with elevated ammonia. This is just to show you that larval zebrafish at 4 days of age exhibit a hyperventilation in response to either 250 micromolar or 500 micromolar ammonium chloride. We know that the neuroepithelial cells are ammonia sensors in zebrafish as well. This is work done by master student Sarah Abdella. What she's done here is intracellular calcium recordings of single neuroepithelial cells exposed to high ammonia. And here are the quantified data here. I'm only showing the ratios, not the absolute data, but clearly there's a calcium response. So to answer the question, are Rh proteins involved, we adopt the technique which we use in our lab very often, and this is gene knockdown, a translational gene knockdown using morpholino technique. And what we do basically is inject antisense oligonucleotides into single cell embryos. We target these nucleotides at the untranslated region on the 5' end, just at the area of the start codon. And of course, this prevents translation of protein. And you can see how effective this is if you compare wild-type fish versus morphins. Morphins are fish injected with morpholino. You can see that in all of the morphins here, there's no expression of the Rh protein when they were injected with morpholino. In today's talk, I'm going to focus exclusively on RhBG and RhCG1. So are the Rh proteins involved? The answer is clearly yes. Here's the normal ventilation response in a sham fish. Here's a fish experiencing RhCG1 knockdown. And similarly, the response is blunted in a fish experiencing RhBG knockdown. So the question that becomes then, can we show that the Rh proteins are localized in an appropriate way to allow this ammonia detection to occur? So the green dots here refer to cells that are expressing RhCG1. The pink cells refer to the neuroepithelial cells, and they're expressing serotonin. And if you look at this at high magnification, what you can see is there's absolutely no colocalization. These neuroepithelial cells do not possess any RhCG1, even though that was our original prediction. So RhCG1 is not localized on apical membranes of neuroepithelial cells. Where is it localized? Well, in fact, it's localized on a group of cells that are positive for the lectin concanavalin A. And we now know that these cells are ionocytes. So the RhCG1 shown here in green is localized to the apical membrane of a specific ionocyte, which is an ion transporting cell known as the HR cell. What about RhBG? Well, RhBG is also localized to these concanavalin A positive cells. Concanavalin A here shown in blue, and if you just focus on this single ionocyte, you can see that it is expressing basolateral RhBG. So RhBG is localized to the basolateral membrane of these same ionocytes. Well, what about the neuroepithelial cells? Here we're focusing on a single neuroepithelial cell stained green. The red refers to RhBG protein. And what you can see is that among the other cells, the neuroepithelial cells are also associated with RhB staining. And that RhB staining is localized to the basolateral membrane. So with this in mind, we've generated the following hypothesis: that the neuroepithelial cells are able to detect ammonia because they allow ammonia to, actually, this is the neuroepithelial cell here, externally applied ammonia finds its way into the neuroepithelial cell through a rather circuitous route. It first has to go through the HR cell or perhaps other cells on the skin of the fish, including keratinocytes. It then exits the ionocyte through the RhBG and enters the neuroepithelial cell through the back door, if you like, entering through the RhBG protein. Any ammonia which is accumulating in the blood because of metabolism or after a meal will enter the neuroepithelial cell through the RhBG protein. So that's the current working hypothesis. What about hypoxia-inducible factor and its role in controlling breathing? We know that hypoxia-inducible factor is a master transcription factor. It's stabilized under hypoxic condition, so it's post-translational stabilized, and it activates all sorts of genes associated with energy metabolism, O2 uptake, delivery, and cell survival. The question that we've been asking in our lab, and this is work done by postdoc Milica Mandic, is does hypoxia-inducible factor promote the hypoxic ventilatory response? And in mice, there's some evidence of this. In a partial knockout of HIF1 alpha in mice, there's a diminished hypoxic ventilatory response. But you cannot get a full knockout in mice. It's lethal. We also know that the hypoxic response is blunted in HIF1A knockdowns in 4-day-old zebrafish larvae. And here are the data shown here. This is work done by Vili Tsunava. Here's the typical ventilation response in a hypoxic sham larva. And here you can see the large blunting of the response if the fish is no longer expressing hypoxia-inducible factor. Well, we wanted to turn to gene knockout And so in our lab, we've been able to work successfully with 2 knockout lines. There are 2 paralogs of hypoxia-inducible factor in zebrafish. There's HIF1 AA and there's HIF1 AB, and we have knockout lines for both of them. And also, Milutz has created a double knockout, so she's bred these 2 fish together and obtained a double knockout. The data I'm going to show you now refers only to the fish experiencing knockout of HIF1 AB. What's very interesting, if you look at the response of a wild-type fish at 4 days post-fertilization to hypoxia, you see that they hyperventilate, but they do so in an extremely transient manner. Hyperventilation peaks at about 5 minutes and then rapidly returns back to normal. But as you extend development from 7 days to 10 days to 15 days, what you see is that the duration of the hypoxic hyperventilation gets longer and longer. So the hyperventilation is not sustained throughout exposure until 10 days post-fertilization. What if you compare the wild-type zebrafish with the knockouts? Well, what you can see at 4 days, the effect is completely obliterated. There's no longer hyperventilation in the knockouts. At 7 days, the effect is greatly reduced, as it is at 10 days. But at 15 days, there's no longer an effect of knockout. So the knockouts are blunting the hypoxic ventilatory response until 15 days fertilization, suggesting that until 15 days, clearly hypoxia-inducible factor is important, and perhaps at 15 days, other pathways are coming online, giving redundancy to the system. Well, what about adults? In adults, there's also no effect of knockout of any of the HIF1 alpha paralogs, or the double knockout for that matter. Fish exposed to progressive hypoxia hyperventilate in exactly the same way. So it's likely that there's redundancy in control mechanisms that means that hypoxia-inducible factor alone is not required to promote a hyperventilatory response. But what about the mechanisms? How could hypoxia-inducible factor be promoting hyperventilation? Well, in 2015, Cosima Porches published a paper showing that nitric oxide stimulates breathing in larval zebrafish. We know that nitric oxide synthase, the enzyme that makes nitric oxide, has hypoxia-responsive elements associated with it. So we thought it was possible that hypoxia-inducible factor could be working through nitric oxide synthase, activating nitric oxide, and thereby stimulating breathing. So what we did was we relied on an inhibitor of nitric oxide synthase, LNAME. And so just here are responses again that you've seen before at 10 days post-fertilization. Here's the wild type hyperventilating. Here's the HIF1AB knockout, a very diminished ventilatory response. What if you take a wild type and you prevent it from making nitric oxide? Well, what you end up with is a response that looks remarkably like the HIF1 knockout. So if the fish can no longer make nitric oxide, we're basically copying the phenotype of the HIF1 alpha knockout, suggesting that nitric oxide may be a key mechanism in allowing hyperventilation and working through HIF. So this image is meant to remind me to look at the time. We started late. If everything had gone correctly, we should be at about 20 to 1 right now, and I've got us about 3 minutes after. So I had to make a decision if we were before 20 to 1 or if we were after quarter to 1, would I continue or not continue? So if I actually click on this clock, the presentation ends. And if I click off the clock, I continue for about another 5 minutes. So I'm going to continue. Okay, so the final thing I'm going to talk about in the next 5 minutes is the role of serotonin, or 5-HT, in chemoreception. And through work done by Sarah Abdallah in my lab and other labs throughout the world, a basic model for signal transduction has been designed, if you like, or formulated for the neuroepithelial cell. And here we're seeing the response to CO2, but many of the properties could be shared with oxygen or ammonia. And the take-home message here is that when the neuroepithelial cell is stimulated, a potassium channel, a background potassium channel, is inhibited. This results in depolarization of the plasma membrane, which results in the release of calcium from intracellular stores. It's very interesting. Unlike the carotid body, there's no calcium entering from the extracellular fluid. All of the calcium is coming from intracellular stores. This results in the mobilization of vesicles containing neurotransmitter, which we believe is serotonin, to the membrane. The neurotransmitter is released and interacts with afferent neurons to evoke hyperventilation. And so the question we've been asking is this neurotransmitter truly 5-HT? Not a particularly easy question to answer. And I'm going to show you some work that was done by a master's student in my lab, Gregory Jensen. And we decided to exploit the synthetic pathway that brings tryptophan to serotonin. And we focused on the rate-limiting enzyme in this pathway, tryptophan hydroxylase. So tryptophan hydroxylase converts tryptophan to 5-hydroxytryptophan. Well, if you work with zebrafish, you know that it gets a little complicated because there are numerous paralogs of many genes. TPH has 3 paralogs in zebrafish, TPH1A, TPH1B, TPH2. But what we've done is we've developed knockdown techniques for each one of these paralogs, and we've also developed knockout lines for each one of these paralogs, which we're currently working with. I'm not going to show you any of the knockout data today. So we've used these knockdowns to inhibit or to prevent tryptophan hydroxylase from functioning by preventing its translation. And the key enzyme in this story is tryptophan hydroxylase 1A. So this is some immunocytochemistry of the back end of a zebrafish, and these red fibers are nerves, and the green refers to TPH1A immunoreactivity. And I'll just blow this up for you right away. And what you can see here is that these peripheral nerves are staining positively for the enzyme tryptophan hydroxylase 1A. So these are serotonergic neurons. The question is, are these serotonergic neurons actually innervating neuroepithelial cells? Well, the answer is yes, they are. And I think I'll just go immediately to high magnification and draw your attention to this cell on the right. This pink cell is a neuroepithelial cell staining for serotonin. You can see nerves here impinging on that neuroepithelial cell, which are showing positive immunoreactivity for TPH1A. So yes, indeed, the TPH1A is expressed in peripheral nerves that are innervating the neuroepithelial cells. And what's very interesting for us is we never saw TPH1A inside a neuroepithelial cell, but only associated with these serotonergic neurons. So then we predicted that knocking down TPH1A would prevent the accumulation of serotonin, because we began to think that serotonin is getting into the cell by its movement from the nerve through a serotonin transporter into the neuroepithelial cell. So what these data show you is a control fish or a sham fish, and each one of these green cells is a neuroepithelial cell showing serotonin immunoreactivity. But in the knockdown fish, you see a marked reduction in the number of detectable neuroepithelial cells. The cells are still there, they just have no serotonin in them, so you can't detect them. And so you can see that there's a massive decline in the number of detectable cells in these TPH1A morphins or knockdowns. And the question is then, what can the fish still hyperventilate? So here you see the sham fish exposed to hypoxia. it shows a pronounced hyperventilation. But the morphant, the hypoxic response is absent. So they're no longer able to hyperventilate if the neuroepithelial cells can no longer accumulate serotonin. So with this, then, we formulated the following model. The neuroepithelial cells, we believed, are innervated by serotonergic efferent neurons that contain TPH1A, allowing the synthesis of serotonin. We believe that the serotonin is moved into these neuroepithelial cells through the serotonin transporters, of which in zebrafish there are two paralogs, SERT1A and SERT1B. Serotonin is then moved out of the cell during activation, but we also think the SERTs are playing a role in bringing serotonin back into the cell. And we're testing these hypotheses right now with PhD student in my lab, Kevin Pan, both with the knockdown and the knockout models. And basically what he's trying to show, first of all with pharmacological techniques, is that treating fish with serotonin transporter inhibitors such as the antidepressant fluoxetine will prevent the accumulation of serotonin in the cell. So with that, I have some final words. I hope the audio is going to work, and I hope if I click on it, it will work. I was told by Oliver and the Christine, the president, that there would be no questions. So I thought I would just let you hear these words.
I'm not going to give you a question. I'm not going to give you a question. I'm not going to give you a question. I'm not going to give you a question.
So, as Donald Trump said, I'm not going to give you any questions. But I do want to thank you very much for listening. I'm sorry if I went a little over time, and I believe it's time to have lunch. So thank you very much.